Welcome, dear students. This is Dr. Adi here, and you are watching Medical Globe. Today, I brought the topic from biochemistry, the properties of monosaccharides. This will be the third lecture from the properties of monosaccharides. Today, we will discuss some of the reactions of monosaccharides, like enolization or tautomerization. You can write this on a page. Number 2, reducing properties of monosaccharide. Number 3, oxidation of monosaccharide. Number 4, reduction of monosaccharides. So these 4 reactions will be discussed today. Let's start with the first one, enolization or tautomerization. In enolization, a compound called enediol is formed. How? Come here. If you take 3 different monosaccharides, or if you add glucose in alkaline solution for 2 to 3 hours or 4 to 5 hours, what will happen? Glucose will convert into mannose and fructose. This is number 1. Or you take 3 monosaccharides like glucose, fructose and mannose. The structure of the glucose is not fully mentioned. However, only 3 carbons are mentioned. The remaining carbons are represented with R. So as we already know the structure of glucose, this is carbon number 1, C double bond OH, aldehyde functional groups. Carbon number 2, 3, you can add H on the right side, NOH on the left side, and then remaining carbon number 4, 5th, 2nd are all same, HOH, HOH and CH2OH. Similarly, fructose, carbon number 1, CH2OH, carbon number 2, C double bond O, and then remaining carbon you can write with the HOH, HOH, right? You can move on. Similarly with mannose, C double bond OH, OH on its left side, HOH, HOH, HOH. All of these are monosaccharides. These monosaccharides undergo a kind of reaction called enolization or tautomerization. What is meant by tautomerization? Tautomerization means the transfer of an atom or hydrogen atom within a molecule from one carbon to another carbon. From one carbon, you will get this easily in the example here. First, come to the glucose. In glucose, this hydrogen, which is attached here at carbon number 2, this whole hydrogen will come to the oxygen, because this oxygen is highly electronegative, so the double bond will shift towards the oxygen, and as a result of this, oxygen will get a negative charge. Then this hydrogen will leave its bond. The bond, because this carbon has how many bonds now? 1, 2, 3 bonds. The 4th bond, which was a pi bond, has been shifted towards oxygen atom, and now this carbon is electron deficient. So what will happen? This bond between the carbon and hydrogen, this bond will come here, and in between this carbon number 1 and carbon number 2, it will adjust here, while the hydrogen atom, it will be shifted towards the oxygen, and a structure like this will be formed. Hydrogen will come here, okay, this hydrogen moved, and this bond will shift here. Now everything is okay. Now carbon has got now 1, 2, 3, 4 bonds. This carbon has also got 4 bonds. Now you see a double bond has been created between carbon number 1 and carbon number 2. Therefore, it is called as ene for double bond, ene, like ethene, propene, okay. And diol, because at the two carbon in between which there is a double bond, these two carbon contain two OH groups. Although we have so many OH groups, but the two carbon in between which we have a double bond, these two carbon have two OH, so therefore we say diol. Diol means two OH. So glucose become like this. Glucose become like this, enediol. Glucose has formed enediol. Look at that, carbon number 1 and 2 has a double bond in between them, OH, H, OH, and then so on. This is not called a glucose now, this is now called as enediol. And remember, glucose can form enediol if it has a free aldehyde group, Now it is possible that glucose can be a reducing sugar. Some students ask that how glucose is reducing sugar. So my answer is, this is the reason that glucose can form enediol, and this enediol can reduce other compounds, means this is highly reactive. It can give electron to others, means it can reduce others, so it can act as a reducing. That is why we say the reducing property of monosaccharide. All monosaccharides have the ability to reduce some other compound. All monosaccharides can act as a reducing agents. The cause is because they can make enediol. Any sugar that cannot make enediol, any sugar they cannot form enediol, will not act as a reducing sugar. So all monosaccharides are reducing, because all monosaccharides, whether it is glucose, whether it is a fructose or mannose, they all can. If you see fructose can also do the same. Means if you see the double bond over here and one hydrogen, this hydrogen, because we have two hydrogen here, right? Two hydrogen. So there will be same mechanism. One hydrogen is here, one hydrogen is here. One of the hydrogen will be shifted completely to the oxygen, right? Okay, this is completely shifted towards the oxygen and will become OH, while the bond will be created as like this. So once again, what happened? Again, enediol has been formed. You can write this H here. Okay, enediol. So fructose can also make enediol. This is mannose. Again, what will happen? You can write it again that this double bond O will be shifted towards oxygen, and oxygen will become electronegative. Carbon has got 3 bonds now. What will happen? This hydrogen will shift towards the oxygen, so it will become OH, and the bond will come here, right? It will become like this. So now we got again enediol. So any monosaccharide, if you put them in alkaline solution, look at the alkaline solution is necessary. The medium must be an alkaline. In an alkaline medium, these reactions are possible. So whatever monosaccharide you add, they will form enediol. The symptom of the enediol is there is a double bond between carbon number 1 and carbon number 2. This enediol can act as a reducing agent, and it can reduce other. So now we will go to a laboratory test, identification test for sugar. That how we identify sugar. Any sugar, if it has a potential aldehyde group or ketonic group, both. Like glucose, we have a potential aldehyde group, free aldehyde group is present. In fructose also we have a free ketonic group is there. So these carbon aldehyde or ketonic carbon are responsible for the reducing action, because these can make enediol. And one of the most interesting thing is sucrose is not a reducing sugar, although other disaccharides like maltose, like lactose, they are reducing, but sucrose is not reducing. What is the reason behind this? Sucrose is not reducing because if you see sucrose, in sucrose there is a glucose, right? There is a glucose and a fructose is there, right? It is a fructose, one fructose, one glucose. Now if you see here, in case of fructose, in case of, look, I'm writing F for fructose and G for glucose. In case of fructose, the carbon number 1 is outside the ring, while carbon number 2 is inside the ring. In case of glucose, this is carbon number 1 and 2. We have already discussed that in case of glucose, the carbon number 1 is the carbon having aldehyde functional group. So this carbon was responsible for the formation of enediol, and it is now busy. This carbon has made a bond with the fructose. So we cannot expect this carbon to make enediol now because it is now engaged, it is not free. In case of fructose, the carbon number 2 was having the double bond O. So in case of fructose, the carbon number 2 is the carbon which we expect that it will make an enediol, but this carbon number 2 is also engaged. The two carbon both are engaged, and hence we cannot expect that the sucrose will make an enediol. Sucrose cannot make enediol, so sucrose is not a reducing sugar. That is why we got the very special answer why sucrose is not a reducing sugar, because it does not have any reducing property. It does not have a free aldehyde or free ketone group. However, if you hydrolyze sucrose, so what will happen? The glucose and fructose, this glucose has its free aldehyde carbon, fructose will got its free ketone carbon. Both the two carbon will be now available for the reducing action. These both will make an enediol. So after hydrolysis they will become reducing. Fructose is reducing, glucose is reducing, but once they make bond with each other, they will no more reduce other compound. That is why it's a very special thing. All monosaccharides are reducing, all disaccharides are reducing, except sucrose is not. Even polysaccharides are also reducing, because they make there will be so many monosaccharide link, but with the first and the last monosaccharide you may see the carbon, free aldehyde carbon or free ketone carbon. That's why. So now the reducing properties of monosaccharides, the laboratory test. Whenever you perform a laboratory test for identification that contain copper sulfate, a green color compound, copper sulfate, when you add copper sulfate in water, you will get copper plus ion. This is called as cupric. This is called as cupric. Cupric. It has double positive charge. It has lost 2 electrons. And what will happen? Now what will happen in the laboratory? When we add a sugar, means any monosaccharide or disaccharide except sucrose, the sugar will make, or it will become enediol. It will become enediol. And we know that enediol is very reactive, so enediol will give electron to the copper. Copper is plus double positive charge, so it give electron to the copper become Cu positive 1. There were 2 positive charge, now it become 1 positive charge because 1 electron has been given, so it has now Cu only 1 charge. So a kind of reduction occurred here. The copper got electron, so now it is called as cuprous ion. It is now called as cuprous ion. This cuprous ion will be in the form of cuprous hydroxide. It will be yellow color. If you further heat it up, it will release water and it will become Cu2O. This is called as cuprous oxide. This is called as cuprous oxide. It has red color, and cuprous hydroxide has yellow color. So the color, the presence of color yellow or red will indicate that there was sugar in the sample, because in the laboratory test we just take a sample and we just want to check either it contains sugar or not. If it contains sugar, it must form an indiol, and if it make indiol, it must reduce copper sulfate. So this is the basis of the identification test for a carbohydrate, whether you take Benedict's test, whether you take Felling's test. Okay, so Benedict's test, Felling's test, these are all are based on this indiol. So whenever you are standing in the lab, in the biochemistry laboratory and the lab, you are performing this test, the principle of this test is all linked to this indiol formation. So that was very important for me to tell you. And now after reducing properties of monosaccharide, we will come to the oxidation of monosaccharides. The third property, this is first, second was done, second was the reducing property, and the third is oxidation of monosaccharides. Oxidation of monosaccharides. Okay, can we oxidize monosaccharide? So the answer is yes. You can oxidize monosaccharide. If you got a glucose, let's suppose it's a glucose, and glucose contain C double bond O H, and then you can write second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. Sixth carbon is so easy to write, CH2OH. Other carbon you will write OH on the right side, OH on the right side, except the third one where you will write OH on the left side. So wherever you have mentioned OH, on the opposite side you will mention H. Now this is H. Okay, so as a D-glucose, because the OH is on the right side, there is now D-glucose because the OH is on the right side. The OH of the fifth carbon is on the right side. It is a D-glucose. You can oxidize it, but for oxidation you can see that you can either oxidize the aldehyde carbon. This carbon can be oxidized, or you can oxidize the terminal alcohol. This is called as terminal alcohol. So what will happen if you oxidize the terminal alcohol? What will happen if you oxidize the terminal alcohol? Then this will become glucuronic acid. This will become glucuronic acid. Glucuronic acid means you will get C double bond OOH here. C double bond OOH here. How? Like this. COOH. The whole compound will be like this. If COOH comes here, then this compound will be called as glucuronic acid. Glucuronic, right? If you oxidize, means CH2OH. If you oxidize the carbon number 1, means aldehyde carbon, it will become like this. COOH, means if COOH comes here, everything will be same, then it will be called as gluconic acid. Gluconic acid. So gluconic and glucuronic acid, small difference is there. If aldehyde carbon is reduced, it will be called gluconic acid, and terminal alcohol, it will be called as glucuronic acid. COOH, you can write it as well like this, C double bond O and OH, right? Look, there was C double bond O and hydrogen, but after oxidation you can see an extra oxygen there. It shows that oxidation has occurred. Now these compounds has a special role, gluconic acid and glucuronic acid. You will see these glucuronic acid in different kinds of mucopolysaccharides, heteropolysaccharides. They are physiologically very important monosaccharides, and it will help you understand the polysaccharides. Now the last one is the reduction. The last property is the fourth property is the reduction of monosaccharides. You can reduce monosaccharides as well, but for reduction we will again take the example of glucose. You can take glucose means D-glucose, because, and you can take D-galactose, and even you can take mannose, D-mannose. You know, these are all isomers, glucose, galactose, mannose, all are isomers. You know very well, already we have discussed this in the topic of epimerization or epimers. If you reduce the glucose, for reduction you will use sodium amalgam. Sodium amalgam is the reducing agent, sodium amalgam, and you will get what? You will get D-sorbitol, and if you reduce D-galactose with the same agent, sodium amalgam, what you will get? Dulcitol, dulcitol, and D-mannose with the same, you will get mannitol. All contain ol, means if you have here C double bond O H and then carbon, every carbon has OH till the last carbon, it also contain OH. But reduction means that at this carbon you will get one more extra hydrogen and the O become OH. Now this is a monosaccharide, but you will not see double bond O, you will see OH and H. So we got how many? 2 hydrogen here. It was C double bond O and H, remaining carbon were there. So what happened? You need to give 2 hydrogen in reduction. 1 hydrogen will come here and it will become OH, other hydrogen will come here, it will become CH2. Now this become an alcohol, complete alcohol, not aldehyde. So you can reduce glucose. If you reduce glucose will become D-sorbitol, galactose will become dulcitol. You can write the structure of all these sorbitol, just make it like this, everything will be same. Sorbitol and dulcitol are clinically very important because they can cause, if they accumulate inside your cell, they will damage your cell, they will cause inflammation, and they will cause as a nephropathy, different types of nephropathy. And this one, mannitol, is also important, these two cause. Okay, I think we should end up here. The light has almost gone. Mannitol we will discuss in the next video. Mannitol is used as a forced diuresis and to reduce intracranial pressure. The light has gone, so I hope the lecture was also ended. Thank you and see you. Bye bye.